Welcome to this quick tutorial on SNPversity 2.0 in grain genes. SNPversity is a web-based tool for exploring genetic variation. Currently, the platform hosts wild barley data, with additional datasets planned for the future. In the next few minutes, I'll show you how to select a genomic region, choose accessions, view variants, download a VCF, and build a phylogenetic tree. Let's begin. Step 1. Select a genomic region. Start in the Select Options tab. First, choose your genomic interval. You have two options. Option 1. Select a chromosome, from 1H through 7H, and enter a start and end position in base pairs. Option 2. Enter a Barley-Morix version 3 gene model ID and click Load Coordinates. The chromosome and positions will auto-fill. Regions larger than 1 megabase are available for download only. Table and tree views are limited to 1 megabyte or smaller. Step 2. Choose a dataset. You can choose high-quality SNPs or indels, high-coverage SNPs or indels. High-quality datasets include additional filtering, including linkage disequilibrium. High-coverage datasets include more variants. Select your dataset by clicking the checkbox. Step 3. Select accessions. Now choose which accessions to include. You can select them manually, select all, randomly sample a percentage, or upload a file of accession IDs. Each selected accession will appear as a column in the results. When done selecting the region, dataset, and accessions, press the Create and View the VCF file button to generate the selected data. Step 4. View the table. Click the Table View tab. For regions under 1 megabyte, you'll see a color-coded table. Each row represents a variant position. Genotypes are coded as 0 homozygous reference, 1 heterozygous, 2 homozygous alternate, N missing data. Variant effect types and impacts, predicted by SNP effects, are provided for each SNP. Pressing on the associated gene models will open a new screen, showing the selected gene on the genome browser. Clicking on the Download the VCF file button will download the file containing all variants in your selected region and accessions. Step 5. Build a phylogenetic tree. Open the Tree View tab. Choose a UPGMA tree or a neighbor joining tree, and the type of output format you would like to generate. Press Draw Tree to generate the selected results on the screen. SNPversity 2.0 makes it easy to explore genetic variation data directly in your browser. Additional information is provided in the Downloads and Help sections. To get started, visit link to graingenes.org. Thank you for watching.